I'm the co-founder and CEO of Nova, which is specialized in the emerging field of in silico clinical trials. And emerging is actually the operating word here. It took 2.5 millennia for the randomized placebo-controlled design for phase 3 trials to emerge and establish itself as the gold standard. From the first controlled experiments to the landslide regulation by the FDA in the middle of the past century. Now, fortunately for us, in this digital age, things are moving much faster. Both the EMA and the FDA are advancing regulatory science in modeling and simulation. This letter from the FDA's Modeling and Simulation Working Group, and it states that in silico clinical trials has become a top science priority for the agency. And actually, the latest appropriations bill voted by the US Senate a couple of weeks ago has a full paragraph dedicated to in silico clinical trials. Now, zooming in on Nova, we are a professional services company. We're supporting our biotech and pharma partners in optimizing their preclinical and clinical development strategies. We achieve this thanks to a proprietary approach which enables us to predict the clinical benefit of a drug candidate in silico, potentially long before the first trials on humans. We have established market traction from preclinical advisory services down to real-world evidence value demonstration, and it essentially serves to explore a wider range of assumptions, cheaper and faster than would otherwise be the case with more conventional in vitro and in vivo assays, improve probability of success, so therefore de-risk, and ultimately support decision-making throughout the entire process. Our magic formula is called the effect model of a drug. It is computed in a very simple two-step process. First, we apply a mathematical and computational model of the disease of interest to a virtual population of patients representing a real geography and context in order to get the risk of the clinical event of interest without treatment, which is represented here on the y-axis. Then we add a drug submodel to the entire disease model itself, apply this back to the same virtual patients in order to get the risk of the same clinical event of interest, this time around modified by the treatment, and this is represented on the y-axis. Now, the further away the patient sits from the line bisected in this plane, the larger the expected clinical benefit from the drug candidate. And this gives us, for each patient, what we call the absolute benefit. And if you sum this over the entire population, you get a population efficacy metric we call the number of prevented events. Applying this to an mTOR pathway inhibitor in the prevention of long graft rejection, we were able to profile the optimal responders sitting over there and reduce the size of a phase 3 trial from 3,000 patients to 600 patients, which would generate potential savings of $70 million. Now, there's a lot of hype around big data and machine learning these days, and overhype and inflated expectations tend to kill nascent industries. Warren Buffett, the famous investor, was quoted saying, "It's only when the tide recedes that you get to see who's swimming naked." So let's step back for a second and try to understand why using data uniquely as a base material for modeling purposes makes for suboptimal predictive capabilities. Those models are based on correlation analysis. Data is extremely time and context dependent. Most of the data sets available are collected in observational context and not in the carefully designed clinical trial setting with a control group, and the algorithms operate as black boxes. Our approach is much more reliable in terms of predictive capabilities. We basically build those disease models based on knowledge extracted from the scientific literature. Those are basically mapping all the biological entities involved in the mechanism of interest, and we map with and represent with mathematical equations the functional relationships between those biological entities. Plus, our technology platform operates in a full white box manner where you can actually trace back from the simulation outputs to the primary source of information. Thank you for your time.
Thank you. We'll follow with questions from the audience. Sorry, not the audience, the jury first, sorry. Who wants to go first?
Can you help me? I miss what you protect actually, because in the last couple of slides you very clearly outlined that you use data which is available in the public domain, and that you have a white box approach, which is good on a lot of point of view. But what do you protect and how then?
Well, basically, if you picture a disease model, the bulk of the information we need to build the model is available in publicly available sources. But then, when we partner with biotechs and pharmas, we will get access to their own internal knowledge they've generated, for instance, on the mechanism of action of their drug, and potentially proprietary data that we will use, in part, to calibrate and validate the predictive nature of the model. So in other words, there is the large part of the disease model is non-proprietary, but there's a disease or project-specific disease model, which basically corresponds to the specific questions we need to address for the client.
Could I ask a follow-on to that? So with the example that you've given with the mTOR inhibitor?
Yep.
Was this with a partner, and what did they provide in terms of proprietary data?
So this was actually a project funded by the European Commission. We didn't have any industrial partner involved in the project, and basically what we use in terms of external data, apart from those found in the literature, were access to 2 European cohorts of lung transplanted patients that we basically used to develop a virtual population representative of these particular patients.
Okay. Can I ask a follow-on? Just then about how you grow. So if you've done one project here in lung graft rejection, and then do another project with the lung biology program, would the strength of your model improve when there's overlapping biology, or are they kind of discrete consultancy projects?
Well, there's actually a strong multiplier effect within the same therapeutic area, and this is especially the case in oncology, obviously, where you can cover 7 or 8 different cancer types with roughly the same submodels. We usually approach disease modeling by breaking it in fundamental submodels, so you would have apoptosis, cell proliferation, and all of those things. They are largely reusable. So there's a scale-up factor within the same therapeutic area, but to a lesser extent, nevertheless existing, there's also transversal reusability throughout different therapeutic areas.
What differentiates you from your competition?
I guess it's a couple of things. The first one really is methodology. The effect model approach, which is proprietary. The second thing, which is less defensible, is the approach to building the models themselves. The focus on extracting and curating knowledge from the scientific literature, as opposed to only using data as a base material for all the reasons that I've given, which is that it's not really reliable a base material for predictive purposes.
Do you have any other examples on sort of success stories? I mean, here you reduced the trial size, but the trial is still running, right? Or will run?
No, this one is actually an in silico simulation. Okay. It hasn't been validated with a real phase 3.
So and that's going to be the...
That's the GRAL, basically. We've actually filed a request for a Horizon 2020 project. The grant call was called the In Silico Clinical Trials. We should expect the answer within the next couple of weeks from the European Commission, and the primary purpose of this project is actually to perform a prospective validation, run the in silico clinical trial before a real-life one in oncology, and hopefully this one will get funded by the Commission. No, sorry, just to expand and finish on your question, we do have in our portfolio of projects a number of both prospective and retrospective validations in multiple therapeutic areas.
I guess on this project you will have a back and forth discussion with the authorities, with the regulatory bodies, right? So how do you plan to convince them that basically the technologies you are developing will definitely could be accepted by the regulatory bodies?
Well, it's interesting because there's already the common understanding, both at the EMA and FDA level, that data-driven models tend to break. So they really are focused now on multi-scale and knowledge-based representations of disease processes. So from that standpoint, we already belong to the same church, quote unquote. As to the actual methodology itself, the effect model methodology, we're in discussions to secure a qualification opinion from the EMA, which is sort of a vetting process for new methodologies applied to drug research and development. And as I've stated before, the Horizon 2020 grant that we've filed a request for will be done in partnership with the FDA and the EMA. So we'll be interacting with them. They won't be member of the consortium for obvious reasons, but they'll be sitting on the scientific advisory board, and we'll be working on maturing the regulatory science on modeling and simulation with them throughout this project.
How important is the white box feature? Will you go back and dig around? I imagine the scientists would find it interesting, but is it important for the business?
No, it's actually more important from a science standpoint. It's a nice selling point from a business standpoint, but actually making sense of the simulation outputs is obviously critical. So the benefits of this approach is that you can test assumptions, compare the results, and have an easier way to walk back through the entire process and identify potential weak links in the model. I think it was David Barrett this morning referring to the point where we now are in the position to identify the known unknowns. This is also extremely useful from that particular standpoint. We can pinpoint the places where our partner should focus its next in vitro or in vivo assays in order to help improve the predictive capabilities of the model.
Any more questions from the jury? Oh, okay, so any questions from the audience? Okay, can I steal it?
Hope I have understood your model right. Is the result of your modeling influencing the inclusion criteria of a trial? So in other words, is there a potential limitation of the final market for the pharma partner?
Excellent question. In theory, yes. We basically, by identifying those optimal responders that I've shown on the previous graph, in some situations, we would end up in a smaller market. Now, the question is, when you go to the payer with a very clearly specified patient population, it's a trade-off between volume and value. So you might as well be more specific about your target population and then secure a higher price.
And second question, had your model ever resulted in a kind of bipolar or biphasic result, that you have identified two completely different populations for the same modeled drug?
Generally speaking, in most instances, the effect model is curvilinear. There are very few specific instances where it's linear, but those are really particular cases. And actually, it makes intuitive sense. Low base risk patients will stand to benefit less from the treatment because they're less at risk in the first place. And unfortunately, very high-risk base patients without the treatment may be too late for them anyway. So this effect model enables us to really pinpoint those optimal responder profiles.
Thank you.
Any more questions? No? No? Maybe another question from the jury? No? Okay, great. Then
thank you.
big round of applause for